Can you all see my screen?
I think yes. Yeah.
Yeah. Okay. And just checking also. Let me see. Okay, good. So you could, yeah, and the slide transition as well. You can see, right?
Yes. Yeah, we can.
Okay. Excellent. So yeah. Well, thank you so much. For inviting me to present here. What I'm going to do is I wanted to share with you, well, because I'm not, completely familiar with all of the audience, I figured what I'll do is do an initial introduction to just basic microbial communities and using genomics to study microbial communities. And then I'll tell you all a little bit about some of our recent work in trying to understand how microbes interact with each other in these communities and how their interactions compare and contrast across different cohorts and disease conditions and so on. Okay, and I'd be happy to take any of your questions at the end of the seminar. So just as a brief introduction, so the talk is obviously going to be about microbes, and this is sort of a bit of an obligatory slides, and why microbes are interesting, right? I mean, basically the point being that microbes. So when I mean microbes, by the way, I'm talking about the small organisms, typically less than 100 micrometer in size and not visible to the naked eye. And our focus here is really going to be on prokaryotes, and in particular on bacteria, right? And so microbes have existed on Earth for 3 to 4 billion years. They are extremely abundant, right? They've been referred to as the unseen majority of the life forms on Earth. And it's estimated that they constitute more than half of the biomass on Earth. And this is quite incredible, right? Thinking about that more than half of the biomass on Earth we can't really see using our naked eye. Yet they're extremely important. They exhibit incredible amount of diversity. By diversity, I mean, they come in all shapes and sizes and forms, and they can do all incredible chemistry. They are found almost everywhere in extreme environments, environments that are deemed too inhospitable for higher life forms, including us humans, right? They're found in all kinds of temperatures, ranging from -15 to 121 degrees centigrade, range of pH values, high pressure environments, and so on. The study or the understanding of microbial communities from a genomic perspective has really taken off in the last 15 years or so. I mean, of course, studying microbes using sequencing has been around since the 70s. Now I'll talk a little bit about that. But really, the study of microbial communities and the genomic composition really has taken off in the last 15 years or so. And in fact, I've been part of several different projects that have tried to characterize microbial communities in different environments. So back prior to joining UCF, I was at the J. Craig Venter Institute, where we had a project called the Global Ocean Sampling Expedition, where we had circumnavigated the world's oceans, collected water on different size, pumped water through different size filters, and extracted the DNA and sequenced it and tried to study the microbial composition. So the goal of the Global Ocean Sampling Project was to really catalog and understand the microbial diversity in the world's surface oceans. And so that was a project that we continue to analyze data to this day. I mean, we had a whole series of publications back in the mid-2000s, but those data are available to the community, and they continue to analyze it to this day. And I'll talk a little bit more about that. But then since then, a lot of the techniques that we developed, we've brought to bear to apply to study the human microbiome, which is really going to be the main focus of my discussion later on. And we've also looked at microbial communities in airborne environments and in environments like soil and so on. So, one of the big take-home messages from the Global Ocean Sampling Expedition data was this whole idea of diversity, right? That the diversity that exists in nature, we've only begun to scratch the surface, if you, so to speak. And one of the big things that, way back at the beginning of the project that we discovered was that when we collected samples, water samples, and sequenced the DNA from these water samples and compared it to reference data sets, reference genomes, available in NCBI and other locations, we could not get good sequence matches. I mean, basically the point was that we were seeing, as we were generating more sequence data, we were seeing newer and newer parts of genomes of organisms that we had not seen before, or we hadn't cultured before. And there was just a tremendous amount of diversity. We even had this study of, as we do sequencing and generate sequence data, what do these data tell us about proteins and protein families? And one of the big observations was that we continue to identify, more protein families as we sequence all these new environments. And so this was an incredible sort of eye-opener in terms of diversity of microbial communities. And underlying all of that, of course, is, I just want to point out that there's a lot of computation and interesting algorithms that go behind this. And again, the data are out there, and research community continues to analyze these data to this day. Other researchers also have, so for example, from Jill Bansfield's group is a more recent paper where they had looked at water samples from shallow aquifers and also soil samples. And they actually discovered new phyla, which if you're familiar with taxonomy, bacterial taxonomy, to say that we're actually identifying new phyla of bacteria, that is an incredible statement in this day and age, right? And so so the point is that with sequencing technologies, we go to these new environments, we continue to identify an incredible amount of microbial diversity, primarily driven by bacterial and archaeal diversity, okay? Oh, and so by the way, so us humans, we are somewhere in here, right? We're a small leaf in this eukaryotic branch of life. So just a quick word on, how do we study microbial communities, right? And so this whole idea of various omics technologies. And so this idea of, using genomic or DNA sequencing methodologies, right, to study the genomes or constituent of the genomes, right? So one of the earliest approaches was to use what's called the marker gene-based approach, right? So where you target a particular part of the genome of these organisms. In particular, people are focused on the 69S ribosomal RNA gene, for example, or the RECA gene, which provide good taxonomic markers, meaning every bacteria has it. And if you go to an environment, if you're able to PCR amplify, this portion of the genome containing this gene, and then sequence it, then we can use that sequence to build phylogenies and understand evolutionary relationships and the taxonomic composition of the organisms in these communities. And people have also used marker gene-based approaches to study functional genes, right? If you were interested in looking at particular functions in microbial communities. So this whole, marker gene-based approach is one idea. And of course, the field really took off and really overcame this whole cultivation barrier that we have with studying microbes by this whole idea of using metagenomics. Hi, so I'm getting a little bit of feedback.
Can you please, all of you, mute your microphone, please? Yeah, thank you.
Thank you. Yeah. And so metagenomics, I'll talk a little bit more about this now. It's a very simple idea. You go to an environment, you collect samples, be it water samples or skin samples or stool samples, extract the DNA and just sequence it, right? And so the idea then is that you can use computational methodology to study or to make inferences about your microbial community. I'll come back to this in just a bit. So both of these obviously give you sort of the viewpoint from the genome perspective. You have other omics approaches, including metatranscriptomics, metaproteomics, metabolomics, that each look at different viewpoints of the system, if you will, right? So metatranscriptomics helps you look at gene expression levels. Metaproteomics, obviously, proteins and protein expressions, and metabolomics, which is looking at the profiles of small molecule metabolites. Okay, so all of these sort of omics approaches, if you will, give you different views of the same system, same biological system. And it's an interesting, endeavor or effort to be able to integrate these different data types and to come up with sort of a story or make, observations about the system as a whole. Now, just a little bit more on 16S ribosomal RNA gene, just because that for a while was the primary way of studying microbial communities. So this particular gene is roughly 1,500 bases in length. It is found in all bacteria and archaea. It is really interesting because it contains portions of the gene fast evolving, other portions are slow evolving. And so it can be used to build phylogenies and get good resolution in terms of taxonomic classification and so on. And in fact, it was obviously made popular by Carl Woese and his colleagues, Norm Pace and others from the 70s. What has happened since transition from Sanger sequencing to the shorter read technology has been the idea that we don't end up sequencing the entire gene, but we end up sequencing what are called these variable genes. Variable regions on the 16S gene. And it turns out that the 16S gene is 9 variable regions, and different projects over the years have focused on sort of different variable regions, if you will. So in the Human Microbiome project, we'd looked at V1, V3, V3, V5, V6, V9 variable regions. But of course, what's happening now is with the advent of the longer read single molecule technologies like Oxford Nanopore and things like PacBio and all that, you can now start to get back again the full length of the 16S gene. So studying, again, using 16S gene sequencing is an important way of studying microbial communities, right? And what can it really tell you? Well, with sequence data, you can start to then, after you do processing and so on, you can start to get a taxonomic assignment and get a sense of, what are the microbes in these communities. And you can start to do various sort of multivariate analysis with these data in conjunction with all sort of the metadata that's been collected, by these different projects. So 16S as a technology, for studying microbial communities is really popular. It is very cheap to generate lots of data, but one has to be a little bit cognizant of the limitations of 16S. What do I mean by that? So while it's useful for taxonomic classification, to talk about functional relationships between organisms just purely based on 16S can be a bit of a challenge or a danger. And so one has to be careful about overinterpretation. And in this slide, what I've done is I've shown you sort of a metabolic map of 2 different strains of E. coli. Okay, don't worry about what all these different parts of the figures mean. Just from a high level, you can see that these 2 figures are different. These are metabolic maps of 2 different strains of E. coli that have nearly identical 16S genes, but they are very different functionally. So one is actually a human pathogen, the other is not. If you look at the genomic composition, a third of the genome is quite different to each other. And they are different, right? Yet at the 16S level, they're nearly identical. So it is important to observe that 16S does not tell you anything about sort of functional capabilities of organisms. And one has to be careful about over-interpreting 16S data. Okay, and so there are a whole bunch of other reasons as well that one has to be careful when you use 16S data and be aware of sort of limitations. That while it's useful for, while 16S sequencing, gene sequencing is useful for taxonomy and taxonomic classifications, to kind of take it to the next step to really talk about in-depth functional analysis, that is dangerous, okay? And it's not always possible to accurately reconstruct functional capabilities just based on 16S. Metagenomics, on the other hand, of course, as a paradigm is actually quite simple, right? You have a sample, extract the DNA, sequence it, and then you get access to the genomic content of all the constituent microbes, right? And really, there's no need for cultivation of these microbes. Of course, at a high level, what it looks like is you have a bunch of reads, sequencing reads that you've generated from the samples. And then one of the goals really is to bin or identify, where each of these reads come from. So this whole idea of binning, the collection of reads into subsets of or partition of the reads into different organisms. And then being able to do things like assemblies and really honing in on what the metabolic and functional capabilities or the potential of these organisms are, right? And then using that information to study microbial community ecology and function. And so over the years, my group, both here at UCF and previously when I was at the J. Craig Cancer Institute, we've developed various computational algorithms, pipelines, methodologies for analyzing metagenomic data. There are various types of different sort of computational problems, if you will. So this fundamental question of, once you have read data, how do you do relative genome abundance estimation? I'll talk about that a little bit later as part of the rest of the talk. And then other questions like, assembly of the genomes and annotation and sample comparison and so on, right? Ultimately, remember, our goal is to kind of get at this whole idea of genotype-phenotype associations, right? That is, from the genomic data that you've generated and the metadata that you have obtained as part of your sample collection strategy and so on, can you come up with or identify strong associations based on the genotype? So can you build predictive models that can predict what the phenotype looks like based on the genotype information? Okay, so underneath all of this really are a whole slew of interesting combinatorial algorithms and machine learning based approaches as well that people have been, that my group and other groups have been using. Okay. So what I want to focus on for the rest of the talk is really just look at the human microbiome, in particular the human gut microbiome. So why, the human microbiome? Some really interesting stats, right? I mean, so an average human, we have about 30 trillion cells, human cells. And it turns out, there's roughly a similar order of magnitude of cells, microbial cells, that's in contact with our body, in our body, on our body, and so on. Okay. And these microbes and all kinds of, they are important for our survival. When you think about it, as I mentioned earlier, microbes have been around for 3 to 4 billion years. We, human species, we've been around for much less amount of time. And so you can imagine that over the course of our evolution, that the microbes have co-evolved with us, right? Those microbes that are in contact with our body have co-evolved with us. And therefore, it's not surprising that there are certain functions that are extremely important for our survival that microbes provide. In fact, they endow many sort of functions related to immunity, resistance to pathogens, breaking down, different fats and lipids, providing things like vitamin K, a whole bunch of important functions. About 10 years ago, the NIH funded the Human Microbiome Roadmap Initiative, where one of the goals was to really characterize what the microbial communities on different parts of the human body looks like. And so this was an effort that involved collecting various samples, oral samples, saliva samples, stool samples, skin swabs, and so on, to be able to study microbial communities on different parts of the human body. And so one of the take-home messages was that, the community composition is quite different across many parts of the human body. So the gut microbial community is quite different from what you find on skin and so on. And a lot of the techniques that we used for the Human Microbiome project data analysis were approaches that we had designed initially as part of the Global Ocean Sampling Expedition project that I mentioned earlier. Okay. Of course, since then, there's been a lot of interest in study by various research groups out there, including ours, in trying to understand the different microbiomes associated with the human body. For example, the gut microbiome has been an extremely important and active area of research, right? Where it's now becoming a lot clearer that again, the gut microbiome is extremely important for host health, immunity. It's been associated with many different diseases. So for example, it's been observed that the gut microbiome composition in individuals with, say, metabolic disorders like diabetes, or with disorders like IBD and things of that sort, are quite different when compared to the gut microbiome composition of healthy individuals. And so this whole idea of changes in the gut microbiome composition between healthy and diseased individuals, and using is sort of in an early area of research in many of these different disorders and conditions that people are studying. And one of the ultimate goals, of course, is really in trying to understand what is going on, right? What is the contribution of the microbes towards disease formation, disease progression, and how one could use microbial composition and perhaps modulating the microbes to be able to for therapeutic purposes. Okay, and so lots of interesting papers have come out in the recent years to studying understanding what these differences are. That is how what the microbiome changes look like and how they're associated with disease. Now, my take on this in the last few years has been sort of moving on from looking at the genomic composition and so on to be able to study or to be able to use the genomic data to study microbial associations. So what do I mean by that? Well, so let's, if you look at any microbial community, right, be it on the human microbiome or be it on deep subsurface or in deep oceans or whatever, a microbial community structure and composition of the community is really determined by the carbon and energy sources, right? By the food and energy sources that a community has access to. And of course, also by the surrounding sort of environmental parameters like temperature, pH, and salinity, and so on. Of course, in case of human microbiome, these parameters are determined by us, by our host, right? Connected with the host component, of course. Now, the thing to keep in mind, of course, is that in a microbial community, the community members compete with each other for resources. And it's these competitions and interactions, right? It could be either direct, like a microbe A competing or cooperating with microbe B to share food, or there could be indirect interactions where A and B compete or cooperate with each other, and that leads to an impact on microbe C, right? And so the idea is that these factors, together with the competitions and interactions, really determine the abundance levels of the microbes in the community, right? So meaning how successful the microbe is in a community at that point in time is based on the interaction with other partners around it, and also with the environment, right? Now you can think of this as a problem in which, so this whole idea of interactions and associations, you can model using a simple graph, right? Where the nodes represent microbial taxa, and the taxa could be computed at different taxonomic levels, if you will, say genus, a species, or whatever. For our purpose, we'll talk about microbial taxa being at the species level, okay? So you can think of each of the nodes in the graph as being a microbial species, a bacterial species, and the edges representing associations. Okay, so there's an edge between two species if there is an association between them, either direct or indirect. And that's what I mean by an association, either direct interaction or indirect interactions, right? And the edge weight represents the strength of these associations, if you will. Okay, so so this is what I refer to as an association network. And the goal is, can we compute or can we infer from genomic data, meaning either 16S data or metagenomic data, can we infer these networks? Okay, so the idea is that you have a microbial community that you're observing, say, a healthy human cohort, right? You're now collecting samples at say different snapshots, and the goal is from these samples we can of course estimate relative genome abundances. Using that information, can we then tease out what this graph structure looks like? Okay, so that's the goal. Can we infer from genomic data these association networks? And so this is what we start with, these sample taxa matrices, and this is our goal, to be able to infer these networks. Now, one thing to keep in mind is that sequencing data are compositional, right? And so what do I mean by that? That is, we can never get, from the way that experiments are designed and sequencing data generated, we do not get information about the absolute abundances of organisms, okay? it's often, in fact, it's always relative abundances. Unless you sort of spike in known amounts of something and then you can use that to calibrate, you will only end up getting relative abundance information. And that's what I mean when I say that compositional data, that sequencing data are compositional. So what that means is that if you want to do something like correlation, right? If you want to then if you want to compute the weight on these edges and all that using just standard correlation between the different columns, which correspond to the microbial species, well, that's a big no-no in the field, right? Because pairwise correlation can be completely misleading because of the compositional nature. In fact, correlation suffers from this negative covariance problem. And so that's a big no-no. And the other issue, of course, is that the sample size, the number of samples may not be large enough to be able to recover all of these dechoose to edges. And so that makes it for an interesting computational problem as well. And of course, the assumption we impose to kind of deal with this a little bit is to assume that the network structure is sparse. That is, we do not have a large number of edges. So the number of edges in this network is, or in this graph, is only linear in the number of nodes. Okay, and that's a reasonable assumption as well, I think, in from a microbial community perspective, in that it is not the case that microbe A, any given microbe will interact with every other microbe in the community. Often there are physical and chemical barriers to microbial interactions. So having this assumption that any given microbe can have a few number of neighbors that it can interact with, I think that's a reasonable assumption to make. And so there have been several different methods proposed to infer these correlations or partial correlations. And they often all involve one of the first steps is to basically take the raw data here and to do a log transformation. And then you follow this by modeling it using various approaches. The one that we are going to use here is something referred to as a Gaussian graphical model. Okay. Basically, the Gaussian graphical model assumes that the log transform data then follows a Gaussian, follows a Gaussian distribution. That is, it's generated by a covariance matrix of a underlying multivariate Gaussian distribution. And our goal is to identify or reconstruct this covariance matrix. So just high level again. The fundamental idea here is that an edge exists between two nodes if and only if the corresponding entry in the precision matrix, which is the inverse of the covariance matrix, is non-zero. Okay, so remember, our goal is to construct or identify these edges and their edge weights, and really compute what's referred to as partial correlations, because it turns out for Gaussian models, partial correlation is well captured by the precision matrix. Okay. So what I wanted to do now is just briefly talk about one of our recent papers, where we attempted to use this framework to study bacterial associations across 4 different healthy human populations. So we looked at, our goal was to construct and evaluate association networks across 4 different populations. We looked at 4 different data sets dealing with sort of 4 different populations, if you will, American, Indian, European, and Japanese populations. We had about, and by the way, these were metagenomic data sets generated from over 600 fecal samples. We had about 1.6 terabases of data that we analyzed. And our goal was to sort of study these, compare and contrast these different association networks and see how that can be used to sort of understand microbial ecology, right, associated with the healthy microbiomes. So just at a high level, these are the sort of different steps that we took. From the raw data, the first step was to identify or get the taxonomic profile of each of the samples. That is, from each sample, get the relative genome abundances or genome copy numbers. Once you have that, we log transform the profile abundance information. And then from that, we identify, using a machine learning approach, in particular random forest, we identify species or features that can be used to classify the samples in the 4 cohorts. And then we use these features, these distinguishing features, to construct the network. Okay, and the reason we took this approach really was because at this point, you can have several hundreds of species that you identify in samples, and it's just not, given the sample size, it's just not possible to construct accurate networks, and therefore we decided to use those microbial species that we thought were informative from a perspective of being able to distinguish the different groups in order to understand how those microbial taxa or microbial species associate with each other. Okay, so that was the goal. So the first step in estimating relative abundances of genomes, it's a simple process. I mean, we take the reads, we map it to reference genomes, right? And then our goal was to use this information to compute relative genome abundances, or to figure out how many copies of each of these genomes is in the sample. And you can treat this as and the way we solve this is by treating this in a generative framework, and using an expectation maximization based approach that optimizes the likelihood function that's defined in a mixture model, okay, to identify the relative abundances of all the genomes. So the end result of this step, by the way, is for every sample, you get relative abundances of the genomes. So think of each sample as being a d-dimensional vector, okay, where each of the entries in the vector is an integer. And of course, you can turn that into frequencies so that it adds up to 1, okay, proportions. If we do this, and then we start asking this question that is, hey, how are these different microbial species distributed? How prevalent are they across the different, for each of the cohorts, how prevalent are they across the entire cohort? And how many of these organisms are there? You end up with this interesting sort of bimodal distribution. Okay, and this is what this particular graph shows. And there are 4 curves that are shown on top of each other, and they all seem to have the same pattern, which is a bimodal distribution, which seems to indicate the following. There are a small number of microbial species that seem to be highly prevalent in each of the cohorts, okay? Whereas most of the microbial species are not prevalent, all right? So this idea that there is some notion of a, I don't want to call it core microbiome, but this idea that notion of a set of microbial species that are found in each of these different cohorts, right? If we then do something like a principal component analysis, just to see how this sample ordination looks like, from a linear perspective, if you will, when you look at the first 2 axes, do you see any sort of separation of the samples, right? And you can see here that yes, I mean, like the Indian group and the American group, they seem to sort of have their own different, they seem to be sort of more spread out. You have a harder time discerning between like the Japanese and the European groups and so on, right? So the point of this exercise is just to show that PCA in itself does not show good signals for separation of the samples, and therefore identifying what might be some of the taxa or the microbial species that might be used to distinguish between the different groups. And so we ended up using a random forest classifier to be able to learn these 4 different cohorts, right? And by that I mean, we use the random forest classifier to identify distinguishing features, i.e. the microbial species that can be used to separate out these different groups. When you do that, we find that there are about 202 microbial species that taken together can build a really good model with F1 score of 90.96 or so. Okay, so it's really good classification accuracy here. Now, we took those 202 taxa and build the association network using the approach that I mentioned earlier, the Gaussian graphical model-based approach. And this is what the network looks like for the 4 different cohorts. And what I just want to point out here is that just at a high level, and so these are all organized in the same manner. So you have actinobacteria, bacteroides, firmicutes, and then proteobacteria at the end. You can see at a high level that, first of all, there aren't a lot of edges. I mean, with 202 nodes, obviously, the 202 choose 2 edges possible. Certainly not that many in there. But you can also see that the distribution of the edges in these 4 different networks they're not all identical, by the way. So you can see, for example, in the American network, Indian network, you can see a higher density of edges between, say, Bacteroides group. Whereas the Japanese cohort, the European cohort, seems to have a lot more edges between firmicutes, right? So that's the section of the graphs. Well, we took these networks and decided to study them further. And in that paper, we discussed a whole bunch of our findings. And I just want to sort of summarize some of what we found. One is that at a high level, there are differences in the structure of these networks, okay? Positive associations, that is edges that are positively weighted, are more dominant in all of these networks. And also, by the way, we tested to see if these networks shared properties with random networks, okay, these Erdős-Rényi networks. And we found that based on different network characteristics that they are not, they're certainly not random, okay? And we also found that for each of these cohorts, a large percent of the associations are shared with at least one other cohort. That is, cohorts tend to share edges, okay, associations. We also find that most of the positive associations are between bacterial groups that are taxonomically and functionally similar, while negative associations are found between species with the same, rarely found between species with the same genus. We also then identified various sort of features of these networks. I mean, the hubs and the modules within the networks. And so please, if you're interested, you can take a look at the paper where we discuss many of these observations, okay. The similar approach we took to study or compare how microbial associations look like in healthy versus disease situations. And so we have 2 recent publications where we looked at CRC, colorectal cancer, and in IBD, inflammatory bowel disease. And we compared and contrasted how the healthy and disease microbial associations change. And we make a whole bunch of different observations, including, for example, in CRC, there seems to be a prevalence of oral taxing, okay, in the gut microbiome of individuals that have CRC. And we make a whole bunch of observations regarding what might be some of the functional implications and so on. So again, if you're interested, please take a look at this paper. Okay, these 2 papers. The rest of the talk, what I wanted to kind of share with you all is a new sort of framework that we put together a couple of years back, and which I continue to work on. And it's based on the following approach. Okay, so or the observation. So thus far, in the previous exercise, so for example, when we looked at the American cohort, I assumed that there was only one underlying processor or just one network associated with that cohort, if you will. And you can imagine that if you're doing a large microbiome study where you have a whole bunch of sort of various metadata and parameters, range of parameters, and so on, there is no reason to assume that the underlying, sample taxa matrix is only associated with one, underlying stochastic process, right? That there is only one network. In fact, we extend the paradigm and we say that, look, we can, a given sample taxa matrix generated from a large study can be associated with, say, K microbial networks, okay? K unknown networks. And our goal is to be able to infer these networks. And then we developed a whole series of sort of methods to be able to treat or to deal with this mixture modeling problem, if you will. So the number one step is to be able to identify or to come up with a generative model for these count data that we observe. Okay, and so we end up using something called the multivariate Poisson log normal distribution. And we use that to model the counts, all right? I guess I'm gonna skip a little bit. If y'all are interested in the math, we can look over it. But just to let you know that MPLN distributions are basically 2-layer distributions, but the observed count data are generated by D independent Poisson distributions whose means are linked together by an underlying or an unknown multivariate log normal distribution, okay? And so it is if you were to write down the actual equation for it, it looks a little complicated. And it turns out, I mean, this particular density function cannot be computed exactly. And that therefore makes it sort of hard to do things like EM-based approaches in a likelihood framework. And so what we ended up having to do to be able to estimate the parameters in this situation, again, just reminding you all again, our goal is, given the sample taxa matrix, to identify the K underlying distributions that generated these data, right? And so we treat this as a likelihood framework, and then we end up not doing EM because you can't do EM directly because of the complexity involved with the MPLN distribution definition. We end up actually optimizing it using a technique called MM. Okay, in our case, MM stands for minorization maximization. It's actually a pretty simple technique. I mean, the idea here is we have a complicated function, say parameterized by theta. So the function is h, and that's really hard to optimize. What we do instead is we identify this function g, which can be parameterized using theta, and it's a simpler function compared to h. Simpler in what sense? It's simpler to optimize. And it has this additional property that g is a lower bound on h. Okay, and so if you were to then optimize or maximize g, then that will give you a good estimate for h as well. And that's the whole idea behind this MM, or minorization maximization framework that we use here. And in our case, we identify this minorizer function by using, you look at the log likelihood function and use the concavity property of the log function, and we end up using that as the minorizer function. So I'm going to skip over the details. Just, you can the equations and the details, the derivations and all are there in our 2019 paper. So if you're interested, please take a look at that. But I'll just say that you can estimate the parameters using a block update strategy combined with a Newton-Raphson method. And that helps us then optimize, right? Identify the values of the parameter for which the likelihood of the likelihood function is optimized. We also treat this in a sparse network framework. That is, we can solve this using a graphical lasso framework. So we had to extend that approach to deal with this mixture modeling problem. Okay, and so essentially, what do? We evaluated the performance of this method under different situations. Okay, including to look at the effect of sampling, data normalization, and convergence rates, and so on. And again, so I think I'm happy to again go over the details if you folks are interested. But it just suffices to say that these are this method. We see that at convergence, we do require we have a good sense of the amount of data that we require in terms of the convergence parameters as well. And we do find that imposing the sparsity constraint as well helps quite a bit. Okay, and in particular, the MPLN strategy, mixed MPLN strategy, in which we use either cross-validation as a framework or the stability approach to regularization, the STARS framework provides the best performance. We actually then took this to the next level and said, well, okay, are there any real data sets that we could apply this framework to? And indeed, it turned out there is a data, there are lots of data sets out there, but we applied it to this particular data set for this particular study that I was a part of several years back. It was a gut microbiome study from a Malian cohort that included 195 adults and children. And we were looking to addressing this question of, how predictive is the gut microbiome composition with respect to the risk for Plasmodium falciparum infection. Okay, and so we had generated 16S data, so we had count data, and therefore we could model it using the mixed MPLN framework. And of course, we didn't know what the value of K was, but meaning how many networks we needed to look out for. But we solved that problem in a heuristic manner using a silhouette method, right? Similar to what we do for K-means, if you will. And what we ended up identifying were two networks from this data set. And when you dig down into these networks, they actually make quite a bit of sense when you look at the metadata that's associated with these networks. It turned out that this group consisted of individuals in the age group of 10 years or more This was age group of, I think, 6 months or less or so. So there were distinct differences between these different groups. And again, the point being that the bacterial associations in these different groups are quite different as well. So this was sort of a proof of concept, if you will, that then enables us to start looking at what the different networks are and trying to really tease out the biology associated with these microbial associations and how they change between different groups and different disease conditions, if you will. Current work involves developing methods for doing model selection. That is, we don't often know what the value of K is. And so we're trying to come up with methods that will help us determine what the value of K is automatically, right? Using a model selection framework. And so that is some ongoing work. And so I think I'm going to stop here, but I'll just sort of point out in our last slide that in microbiome research, one of the big things that's really happening now is, of course, with the technologies available, we're able to look at or study microbiomes from different perspectives, different omics perspectives. And when you include that with the host data as well, and clinical and phenotype data, now we get into this big picture of sort of, hey, with all these different data types and conditions, we now need to build interesting predictive models that can be useful for us in teasing out the biology, right? And trying to understand host microbiome associations, genotype, phenotype questions, and trying to understand or trying to model disease mechanisms. And that's really been the goal of some of the research that my group does and has been doing in the last few years. And so that's what I'm going to end. And I want to just give a few acknowledgements out to my students, Mark Loftus, Saif, Fasune, and Sahat Abakali, all of whom have graduated and gone on to now do postdocs or in industry. And also would like to acknowledge funding from the National Science Foundation for some of our work on ongoing work on a model selection. Okay, so that's it for me. I thank you for your attention. I'm happy to answer any questions.
Thank you very much, Professor. Okay, so you can just drop your questions in the chat box, or you can just raise your hand, and we're gonna give you the floor to give your question. If you have any questions, please. I'm gonna start. Okay, we have one. I think in the chat box. It's not a question. It's just I would say that neuron rafsome method is also used for optimizing logistic regression supervisor.
Yeah, absolutely. It's used for finding the roots of polynomials, right? And that's a technique that we just used. Yeah, absolutely.
So I'm gonna start with a question first. Talking about those association networks that you presented, is there any way how to study the association, but not just from the taxonomic level, but also from the functional, from a functional perspective?
Absolutely, and thank you for raising that question. I mean, because often one cares not just about the taxonomy, but as you're observing that function, right? Functional interactions as well. And so that's one of the things that we did in our all three of our papers as well. So I would definitely encourage you to take a look at it. But one of the things that we did was we asked this question. Okay, well, so we made the observation that taxonomically the identities and also the interactions between these different species, they seem to be different. I mean, they share a lot of overlap, but there are differences in the taxonomy when you look at the different cohorts. What does that mean in terms of function, right? Are there functional differences between these taxonomic groups, if you will, right? And the take-home message, at least at a high level, is that while taxonomy seems to change quite a bit, there doesn't seem to be a lot of differences at the function level, okay, for healthy cohorts and so on. But of course, when you look at CRC, colorectal cancer versus healthy, or IBD versus healthy, there you start seeing big differences in taxonomy, but also in function. Okay, and the functional groups that are present as well. So clearly, I mean, this idea of linking function with the ultimate phenotype, I mean, that's where one wants to go towards. But the same approaches that one takes to construct an association networks based on taxonomy, you could construct something analogous based on function as well. So where the nodes represent certain functions, and then you could ask, hey, are these functions co-occurring, right, in different settings, if you will. So one could do that. But the approach we took to analyze function was first with a taxonomic direction, and then asking, okay, for these differences in taxonomy, are we seeing differences at the functional level as well for the taxonomic underlying taxonomic groups? Okay, but it's a very good question.
Thank you. Okay, people have any other questions, please? Okay, wait in the ask. I'm gonna just drop another question.
Sure.
It's quite a basic question actually, but I think probably some of us gonna have always this question in mind. Is there any way how to choose the variable region for sequences? I'm talking about the 16S RNA gene.
Yeah. So, we and other groups in the past have looked at this question, right? That is, how good are the different 16S variable regions in picking up certain taxonomic groups, right? So you can ask this question, say the V1, V3 region, how specific and how sensitive it is, right? Or the V3, V5, or V6, V9. One of the observations, and please, I would definitely encourage you guys to take a look at our the Human Microbiome paper. That was one of the PLOS papers that studied the variable regions and the ability of the variable regions to pick up microbes from different parts of the tree of life. The conclusion or the observation that one can make when you look at this analysis, when you do this analysis, what is this, by this analysis, what do I mean by that? I can take a whole bunch of known 16S sequences, do alignment of them, say full-length 16S sequences, do an alignment, multiple sequence alignment, and then just extract out the different regions, the variable regions, and ask this question: hey, if I were to take V6, V9, or V3, V5, how effectively can I distinguish, say, proteobacteria? Or how effectively can I pick up actinobacteria or firmicutes and so on? Unfortunately, there is not a one-size-fits-all. Right, there isn't one variable region that performs equally well in being able to distinguish all different groups of life, if you will. Okay, meaning firmicutes and proteobacteria and actinito and actin on all these different groups. So there isn't a single answer. It's just that it's been so which is why, for example, in the Human Microbiome project, we did evaluate V1, V3, V3, V5, and V6, V9, and I think we ended up going with V3, V5, and V6, V9 or something. So there isn't. So the bottom line is there isn't a particular region that works for every environment and every taxonomic group. Okay, so if you're studying soil samples, for example, you might want to focus on, I don't know, just again, I don't know the answer, but you might want to focus on a V1, V3 as opposed to V6, V9, because that might be the group, that might be the region that helps you distinguish between various Streptococcus or, Acinetobacter groups or something, right? So it is, I would say, unfortunately, it is a context-specific or context-dependent solution, depending on the environment that you study in the community and what you think, what the groups are really involved. Although I should point out that for a given project, I think the last thing you want to do is shift or change the choice of variable regions over the course of the project. If you pick a particular region, stick with it, right? So spend some time and effort figuring out what it is and then what region to pick and then stick with it. Because otherwise then it gets very dangerous. But again, the hope here is that with things like Oxford Nanopore and PacBio and all that, with the longer read technology now becoming more used and the error rates dropping and so on, we can start to get back at the full length 16S sequences again, right? And we should be able to get high specificity and so on. But again, so yeah.
I think in this case, I think the analysis will be pretty different because speaking about the full gene, it's not like full gene. It's not like speaking about just variable regions. Okay.
Yeah.
I mean, is there even the idea of like thinking about I don't know, like some neural network based approach or a machine learning approach that can probably in the future predict the best region in a given context. So at least I'm gonna not spend time in working on specific regions, and there is some other regions that are gonna work perfectly for my project.
Oh yeah, no. So you can certainly start to do this. You can do this now, right? You can take, for example, the collection of all available 16S sequences from RDP, the Ribosomal Database Project, for example, right? There are millions of 16S sequences, full-length sequences. You have labels on them, right? You can build a gigantic multiple sequence alignment. You can align them using a profile-based method if you want. You can extract out this of the different variable regions. And then you can answer this for yourself, right? You can answer this question of, come up with a classifier or a neural network or whatever you want, whatever favorite machinery you have. You can see for yourself which of these variable regions are good. Are they equally good in being able to distinguish or identify, taxa in particular parts of the taxonomic tree, right? You can answer that question. But what I'm saying is that we attempted to do something like that back in 10 years ago or something.
Yeah.
When but the conclusion there were certain taxonomic groups for which you could not for which for example V3 V5 was not good in being able to pull things out, meaning being able to distinguish, right? It's a bit of a trade-off, right? I mean, you might be able to get classification. Remember, even with a variable region, it's very rare to be able to get classification down to the species level.
Yes.
Right? Often you're stuck at the genus level if you're lucky, right? Otherwise, it's usually family or whatever else, right? Species is very rare, which is why, I mean, you have full-length sequences, you can get down to the species, right? So it is, that problem is really context dependent, is what I'm saying. And you can, the question you posed about, can you come up with a neural net? You're in a position to answer that now because you have the data and you can certainly put that together. But what I bet you is that the conclusion that you end up with will be inconclusive in some sense, in the sense that there isn't a single solution. There is no one size fits all. There is no one variable region that will perform equally well for all of the different taxonomic groups in all the different environments and so on. That makes it a little challenging.
Thank you.
Okay.
I think we have a question in that book, yeah.
Yeah, I think so. Did you take and consider diet before sample collection in your colorectal cancer study? So, this particular study, Safiya, was all of the studies that I presented, that I reported on, the cohorts that we used, by the way, they're all data generated from published studies. So in our case, the colorectal cancer work, that was from a Japanese cohort where they had actually looked at late stage CRC and healthy individuals as well, people who had gone in for like biopsies and things of that sort. And so we were comparing associations between the healthy group and the CRC group from that perspective. We did not, and so we were dependent on metadata provided by that group, right? That was published. And there wasn't a lot of diet data. So I completely agree with you that, and I think it's well known in the community also, right? That diet has a big impact on gut microbiomes, right? Necessarily. And of course, things like diet and medication and everything else, right? We didn't have access to diet data, and therefore we could not sort of partition it from that perspective. We could not take that into account. But please take a look at the paper, the CRC paper, to see what the different metadata. So we had things like age and other features as well. So please take a look at that, the gender information and things of that sort too. Okay, but so you raise a very important point, and I think the point being that any of these studies that you want to do, right, as you're building a cohort, it is important to be able to gather not just obviously the biological samples, but all the associated data, right? So you definitely want to have questionnaires where you are asking people about usage, antibiotic usage, diet, and everything else, right? So it is a really big challenge, but that is a challenge that people who are collecting the data should be aware of, and should really be able to then share with the community. Okay. So my the second part of the talk, what I discussed, the multiple network. It has some bearings in what you just pointed out about the diet. So the idea is that if we have a large enough data set in which perhaps we don't have the diet information, okay? Or perhaps you have the diet information afterwards. But imagine you could construct this in we could identify what the underlying multiple networks are in an unsupervised fashion. Then you can go back and link it with diet information and so on. Right, so the framework that I talked about, the mixed MPLM framework, when we get to the point where we have large data sets, we should be able to learn in an unsupervised fashion what might be underlying stochastic processes, underlying networks that are giving rise to the observed data. And then you should be able to overlay on top these metadata, including diet and antibiotic usage and so on.
I think I have another question. Yeah, it's again about the it's quite related to the diet problem. You talked about the neural. I'm sorry, about the association network profiles in different communities, but I don't know if you studied from different perspective, like the population genetic of the community, the climate, the environmental factor, and etc. Like how all those factors influence the association.
So if it were a perfect world, we would have access to all of that information, and then we should be able to compute on it, right? Very rarely when you try to analyze, these pre-published. Again, I just want to remind everybody that we did not generate these data. I mean, I was involved in generating the human microbiome data all those 10 years back at JCDI, but we didn't generate these data. So we were reliant on whatever the groups, collected in terms of metadata. And often, unfortunately, even though there are microbiome standards and so on out there, unfortunately, that all the entries are not filled to the same level of completeness, right, often. And that makes it really challenging, right? So ideally, if it were a perfect world, right? I mean, if you're collecting, building a cohort, you'd want to define your cohort, be able to track them over time, and collect predefined metadata as well, along with all the different time points, right? So that even metadata that you think you may not be using, but perhaps you might, you or somebody else might find useful in the future. But therein lies the challenge there, right? How do we even define something like that? So it's a bit of a challenge, right? When you build these sort of cohorts.
Can I add a comment, please?
Yeah.
Yes.
I just would first, of course, thank you for your great speech and sharing your knowledge with us, absolutely. And I would just share with you the same point of view, because I had a similar study on fecal samples from 2 cohorts, and that was incredibly hard to really have all the criteria we are looking for in this specific cohort. So yeah, specifically if you were like pressed with time, this is like really rare when you find all the criteria you are looking for. This is from a part. Other part, as we know, the gut microbiota is really affected by incredibly a lot of factors, from age, sex, diet, and others really a lot of other factors. What I would ask is that if we are now almost able with all these tools of sequences and stuff like this, are we about to have we can say solution for some problems or some human illnesses through like using antibiotics, using maybe some probiotics or prebiotics. Are you seeing like hope in this just specific way without using any other thing, just with as I've mentioned, antibiotics or pre or probiotics? Yeah, and you
Got it.
know,
Yeah, no.
this is
Thank
it.
you for your
Yeah,
question
thank you.
and your observation. Absolutely. So this whole idea of microbiome-based therapeutics, right? Microbiome modulation to be able. So these examples of at least in like mouse models, right, where mice that have like autism spectrum disorder type symptoms. In fact, this is a study done by Mazmanian's group at Caltech, right, where they'd identified certain toxins produced by Bacteroides fragilis, I think it was, that results in the mice showing ASD like symptoms. And then when you remove that particular bacterium, then it shows less of those symptoms, and it's able to show more of a healthy sort of condition. That sort of speaks to this idea that you can, in certain conditions and situations, one might be able to modulate microbes to be able to help with the host phenotype, if you will. I think that has a lot of promise, but I would also, and there are lots of companies out there that are looking into, for example, using microbiome modulation to deal with inborn errors of metabolism, for example, or trying to come up with, deal with, C. diff, recurrent C. diff infections, right? So using, this idea of prepackaged group of microbes that you want to give as in pills, right, as a probiotic or anything else. But I think those are all worthy causes and good problems to tackle. I think we have methodologies and so on, but this whole the factors you mentioned, diet, age, the fact that we're dealing with dynamic systems, right? And while it's been observed that in individuals, you might be having a particular strain and group of taxa over time, their abundances can go up and down based on what you eat in the morning or in the evening. There's a big dynamic component to this, right? One has to be very careful in how you design these experiments to be able to address a particular kind of question, right? So there's a goal of trying to modulate, or come up with microbiome modulation and microbiome-based therapeutics and so on. What one really has to understand, what are the targets that we want to aim for? And really, is it a taxonomic target or is it a functional target? Really, right? So do we care about, a particular organism, or do we care about what that organism is doing? And perhaps it can be outcompeted by somebody else who does something that's more beneficial to the host, right? I think it's an open space. We have the tools, but I think there's a lot of variance on the data and a lot of sort of still uncharacterized parameters that make it really challenging. I'll give you one example, and I'll stop after that. But one example is, remember, everybody talks about dysbiosis in the gut, right? Everybody uses this term. But I bet you that the definition I have is different from what you have. I may not even have a proper definition. That's the challenge. If you talk to a mathematician, or if you talk to a computer scientist, we are a lot more comfortable when we say we can define the system using a set of parameters, right? When everywhere, if you look at microbiome papers, they say, okay, well, there's gut dysbiosis going on, and it's been implicated in disease X, Y, and Z. But nobody defines what dysbiosis is. I mean, there have been a lot of papers that have come out that have tried to get at that problem about, we think in the gut dysbiosis case, it seems to be an increased amount of proteobacteria and lower diversity in the gut microbial communities. But a lot of that, in my opinion, are quite vague. What would be awesome to have would be, a set of parameters, right? Sort of saying, okay, well, we think that for this diet, this is where we think the healthy microbiome diversity range should be, or this is what the taxonomic characterization, the functional characterization should look like, and so on. We don't have that yet. And so for us to do all of these things, right, where we say, disease is different from healthy, when we haven't even defined what healthy looks like, makes it challenging, right? And we're playing in the space where there's a lot of unknowns, and we're trying to kind of take step by step. And so every step we take, we have to look back and make sure that it's reasonable and we can test it and go back and maybe, fiddle with it some more. And that's what I see where the field is in at this point in time. And one more point that is one that kind of mention is, these methods like random forests or neural nets or whatever, methodologies that are, when you're looking for microbial signatures, right, associated with a particular condition, so disease, CRC versus healthy or something. Often what happens is these biomarkers can be great for, say, classification or distinguishing between the groups. But then when you ask the fundamental question, well, okay, now do those microbes, do we think they have any role in disease progression or disease, initiation? We hit a bottleneck. So this notion of are these signatures good? They might be good from a diagnostic perspective, but they may not be helpful from a therapeutic perspective, right? And that's where even in the computational methodology space, things like explainable AI, trying to come up with models that really useful models that can also make sense of what the signatures would identify. That is also an open space, if you will. The analogy I draw here is between things like, in cancer, for example, right? You have your driver mutations and your passenger mutations. Passenger mutations are going there for the ride, right? The drivers are the ones who are really, making an impact. Same thing. You might have one or two microbial factors that are really driving something, but the other guys are there because they're somehow correlated with the drivers. How do we tease that out, right? These are all sort of interesting, in my mind and opinion. I think these are all interesting open problems still. So sorry, that was a very long answer to the observation and question you made, but I just wanted to get it out there.
Thank you. I think I have just one last question. Actually, it's kind of technical question. You're from a mathematical and computational background, so you talk about a lot of formulas and a lot of equations. But like, let's say if someone from a total biology background and he is not so familiar with computational, how can he study the association like without going so deep in I don't know, random forest or Gaussian distribution or all of that?
Right. I mean, so this goes back to keep in mind, right? We're all aware that the field that we're in, meaning in this case, if it's microbiome research, it's a very interdisciplinary field, right?
Yeah.
Now, obviously, it's near impossible to be experts in every sort of component of that, and that's absolutely fine. But the number one thing is to you need to learn the language, right? So this is what I tell my computer science students, that they should be able to talk to biologists or their biomedical science colleagues. It's important. Same thing with the biologists or the biomedical science folks. They need to be able to speak the language, right? Now, with respect to methods and computational methods and methodology and method development, I would, I think there are two groups of people. One is, if you're a computer scientist or a mathematician and you are a developer of methods, okay, that's good. That's fine. That's important. But of course, when you're developing methods, you have to be careful enough to make sure that you're working on problems that make sense, that have relevance, okay, that what you produce is useful for your downstream colleagues, right? For your colleagues who are in the applied field. But then the second group of people is, I would say, is folks who may not develop methods, but who can be sort of power users of methods, right? So meaning they may not know every last detail about neural nets or random forests or MPLN distributions and all that, no problem. But they need to at a high level understand what these methods do or what the goal is, right? So be able to at least speak the language, okay? But again, they're not designing methods, but at least be able to talk about, hey, the way we're solving this is we put together a likelihood function. We need to optimize it, meaning we figure out the parameter values for which the function is optimized. Why? Because we think that the model, the data generated using this model, right? That whole framework is important because knowledge at some level of the methods then will help you become an informed or a power user, and will also help you avoid getting into trouble when you're applying a method, right? Because the last thing you want to do also is just apply a method treating it as a black box. And if you don't know what the limitations of a method is, and if you're using it, that's dangerous, right? So one of the things, as a big example I would just put out there is, please, if you have microbiome data, sample taxa matrix or something, the last thing and if you're interested in doing sort of co-occurrences or computing partial correlations, the last thing you want to do is compute correlations directly, right? Don't just take the columns and compute correlations. That's the wrong thing to do. And lots of reasons, as I mentioned in my talk, but also lots of papers out there that talk about it. And therefore, what are the things that people do? Well, there are a whole bunch of different methods, some of which I talked about today. So that's an example, right? So I think, and therefore, because interdisciplinary research is a bit more difficult and just focusing on, if you're a computer scientist, just on the CS problem, or if you're a biologist, just focusing on whatever you do in your lab, it's a bit more. It's a lot more rewarding as well, right? Because that's how you make, I think that's how the field makes big progress, right? That is when we bring, and the other big thing is you work with colleagues, right? Collaborators, right? People who teach you and people who you teach as well, right? That's how interactions and collaborations work. You learn from each other, right? So, I mean, that's my take on it. It's been, the fun collaborations have been the most rewarding ones where you learn and you teach, right? Each other. It's always the fun thing, so.
Thank you very much, Professor. Thank you, of course, for your presentation and animation this webinar. It was really a great talk. Thank you very much.
Excellent.
So, do you have anything to add, please?
Nope. Again, thanks for the opportunity, and I'm happy to, if again in the future, if any of you guys have any questions or interested in discussing or collaborative opportunities, feel free to reach out, please. It's always fun to work on some interesting science problems. Okay. So thank you again. Yeah. Take care.
Awi, do you have something to add?
Well, nothing more than just saying thank you very much, Dr. Shibu Yooseph, for the great talk, and I like all the various perspectives, and thanks a lot. And thank everyone for participating. We'll have these put up on ASVCB's YouTube channel pretty soon.
Excellent. Thank you.
Next.
Take care. Yeah. Bye.